Adam is truly a remarkable and unique student, and he finished a master's, BS in mechanical engineering from Northwestern in 2017, and then I think one of the really cool attributes that Adam has is that he didn't immediately go to graduate school after that. He did arguably something much more difficult than that. And for 2 years, he was a 7th grade teacher with Teach for America. So really, honestly, if you want to be challenged, try doing that. After 2 years of exploring that facet of his life, he decided that he wanted to continue his education and started at Rice in the bioengineering department in 2019. And in about December of that year, Adam contacted me and said, hey, I'd like to do a rotation in your laboratory. And I said, absolutely not. I have no interest in having you in my laboratory. Your background makes no sense in my laboratory. You don't know any chemistry. You're not even a bioengineering student. Why did you come here? It really was kind of confusing to me. And so Adam was quite persistent, however, and so he kept on bugging me, and I ignored the fact that I miss a lot of emails and don't respond. I'm sure nobody's ever experienced for me in this room before. But eventually, what I tried to convince him that this was really a bad idea to be in my laboratory. It's like, look, you don't know any chemistry, I teach organic chemistry. I expect my students to know how chemical reactions work. He's like, well, obviously, I'll just have to learn organic chemistry. I'm like, really, you're going to learn organic chemistry while you're a student? But he continued to kind of persist and slowly convince me that this guy really knows how to persist, and he knows how to tackle problems from all kinds of different perspectives. And I got quite excited about that. And so finally, at the very end of February of 2020, I said, you can join my lab. And 2 weeks later, my lab was shut down because of COVID. And I was like, oh my God, I've literally made the worst mistake ever in taking this guy on. Now I have to pay for him, and we're all sitting on arrears doing nothing because of COVID. What are we going to do? But Adam, like many other people who was facing the adversity of COVID, really was able to make excellent use of that time and carried out really a fantastic self-study of a lot of areas that we were interested in. And I increasingly realized that despite all the adversity that we started off with, that this was really going to work out well. Further, after that, Adam applied for the NSF a graduate fellowship and successfully got that. And so my concerns about money went away. Thank you, Adam. I appreciate that. He's really carried out a quite remarkable PhD in that subsequent time. So as many of you know, that my laboratory has been working on self-assembling beta sheet peptides for a very long time. And one of the things that we've been very interested in really for decades is we can make nanofibers, but how can we get those nanofibers to be aligned on a macroscopic scale? And I think that Adam's kind of engineering approach to this solved a lot of problems that we've never really been able to address. And so he's going to tell you about those things in just a few minutes. So over the time of his PhD here, he's published 4 papers. A key paper that he was the author on, and he has 2 really beautiful first author papers that will be a substantial part of his presentation this morning. And after he's done here, assuming that a disaster doesn't happen, which I don't expect, he's a postdoc set up at the Salk Institute in La Jolla, where he's going to shift interests yet again and be looking at kind of neural development and neurodisease there. So with that, Adam, very excited to hear your presentation.
Thanks, Jeff. Hey, can everyone hear me okay on the Zoom? Okay. I'm going to be walking around, so you might not see me, but I'll go ahead and get started. So hello, everyone. My name's Adam Farsheed. I'm a bioengineering PhD student here at Rice. And today we're all here for my doctoral thesis defense titled Fabrication of Macroscopic Nanofibrous Multi-Domain Peptide Hydrogels to Engineer Biological Tissues. So I'm going to take kind of a holistic approach on my background here because, as Jeff kind of alluded to, there's a lot of different things going on in my career, and I want to touch on all of those and hopefully give you a perspective that's very similar to kind of how I view my work and motivates what I do. So I started as a mechanical engineering student at Northwestern, and I got interested in fabrication technologies generally, and more specifically in 3D printing. And I just want to let you know right now, I'm going to throw up a bunch of words and quotes. Those are some key words that I'm going to dive a little bit deeper on into as I go through my introduction. Then from there, I decided, and I'll tell you why I decided, but I shifted into bioengineering and I got interested in the field of tissue engineering, and specifically learned about the extracellular matrix and worked with a class of materials called hydrogels. And then, as Jeff also kind of alluded to, unexpectedly, or maybe like not fully strategically, I stumbled into a supramolecular chemistry lab without ever taking organic chemistry. And to be completely honest, I was pretty scared of chemistry my whole life. And I almost failed chemistry lab in college, but I didn't tell anyone that before I joined the lab. And I worked with a class of materials called self-assembling peptides. So first off, I want to orient us to what is 3D printing. We've probably all heard this buzzword. You may have some experiences with it, but it's basically a class of technologies that consists of layer by layer additive manufacturing. That's in contrast to subtractive manufacturing where you cut away material. Instead, in this case, we're building material. And there are two major benefits that 3D printing offer that I want to highlight. The first being geometric freedom. So with 3D printing technologies, you can begin to build structures that may or not be possible with other fabrication technologies. The second is customizability. And so what that means is when you make a material or you make a structure using 3D printing, it's extremely easy to make a change from that structure to a second print. Now, that's in contrast to a lot of technologies like injection molding or things that use casting, where it's really easy to make 1,000 of the same part, but it's kind of hard to make minor changes from part to part. And so 3D printing, again, is a class of technologies, and I want to quickly highlight two that are important to me specifically. The first being stereolithography, and the reason I want to talk about stereolithography is this is actually the type of printing that I worked on as an undergraduate. And so I'm going to show a quick movie here of how stereolithography works. Basically, you have a liquid polymer vat, and you're going to shine a light at that liquid vat, and usually a photo cross-linking polymerization will occur, and you're able to magically build this solid structure out of a liquid bath. And with these technologies, you can create extremely intricate designs. So I was actually using this technology to build heart stents, which you may be familiar with, are pretty small. And so that's where I kind of started. And the second technology of 3D printing that I want to talk about is extrusion printing. So we are probably, most of us are probably familiar with extrusion printing, but I think of extrusion printing kind of like a hot glue gun that's just on a rotating stage. And so with extrusion printing, there's kind of the classical sense with plastics, but what I'm going to talk about here is extrusion printing using hydrogels, which are softer materials. And so here I have a quick video that's kind of important to me of extrusion printing, which is also called direct ink writing. And what we can see is we're able to kind of intricately build these structures outward. And then as the video continues, you'll see that the structure is starting to be built upward. And one thing that I want to call your attention in this video is these materials aren't solid materials. They're more liquid-like. So their properties are a little bit different from what most materials we deal with. And this video on the right is what convinced me to change into bioengineering. So when I was, after graduation, I went to my first conference, and I was lucky enough to see a talk where there were cardiomyocytes, so our heart cells, that were seated onto a soft hydrogel that was 3D printed, and we're actually watching the beating pattern of these cardiomyocytes, which just kind of totally blew my mind. And so that brought me to my next question when I wanted to kind of continue into graduate school. What's the best application of 3D printing? And I was convinced very, very early on that building biological tissues and the ability to create customization for different people, for example, is a really unique property that 3D printing will offer. So then I'm going to shift gears a little to when I came here to Rice, and that was the first time that I learned about tissue engineering as a general field. And broadly speaking, tissue engineers try to build structures to help replace damaged or diseased tissues in our body, or to serve as replacement organs, or there's a million different applications, but it can be basically split into three different branches. The first being engineering cells, the second being engineering materials, and the third being engineering tissue architecture. So in my work, I focus primarily on the materials and the tissue architecture side, and I want to dive a little bit deeper into that aspect. So if we want to start to build tissues and organs, I think the first question you want to ask is, well, what do cells naturally live in? And this environment is called the extracellular matrix. So that is all of the non-cellular parts of our body are called the extracellular matrix. And I want to quickly highlight some properties of the extracellular matrix that relate to soft tissues, which is specifically the type of tissue that I was trying to engineer in my PhD. So the first, kind of shown on the schematic on the right, we can see a cell sprung out in 3 dimensions, kind of grappling onto these fibers. And that is a schematic of the extracellular matrix, which 1 has a very high water content, 2 is composed of, a lot of times, fibrous proteins such as collagen or elastin, and 3 has this unique mechanical properties called viscoelasticity. And what I want to just quickly mention as I pop this graphic up is, if you think about the tissues in our body, they're not exactly solids, but they're also not exactly liquids. And so they have this, what we call a viscoelastic. There's some aspects that's elastic behavior and some aspects that's viscous behavior. And so on this graph here, what we're seeing is kind of all of the tissues in our body laid out on this spectrum, where we can see that as we go kind of up and to the right, you think of more rigid tissues such as bone and cartilage, but regardless of the tissue type, the extracellular matrix gives this viscoelasticity to our tissues. The second thing that I want to quickly highlight is a class of materials that I became very interested in, which is materials that contain hierarchical anisotropy. So what I'm showing here on the right is skeletal muscle tissue, and you can see the structure is kind of like one of those Russian dolls where there's a fiber inside of a fiber inside of a fiber. And so that's how the tissue is laid out. That's also how our extracellular matrix is laid out in a lot of our tissues. And so this is not only common for skeletal muscle, but you'll find similar sorts of structures in peripheral nerve and even in our heart tissue. So just to summarize very quickly, the extracellular matrix has a high water content. It's fibrous in its structure, and it has this viscoelasticity that I discussed. And together, it's organized into this hierarchically anisotropic structure for the tissues that I'm specifically interested in. And through all of these things, the extracellular matrix is able to provide mechanical and biochemical cell instructive cues. And that's something that I found super interesting about this extracellular matrix, is this idea of a cell instructive cue. And that's something that I kind of push forward with in my work. So I just want to call your attention to a quick study that was done that I got interested in early on in my PhD, as I was primarily working in the muscle tissue space. And can we turn the lights off? Is that just so you can see a little bit? Thanks. Yeah, I think that should be better. So in our skeletal muscle tissue, as I mentioned, there's this aligned structure. And so I'm showing an experiment here where in the first case, we're looking at a cross-section of muscle tissue just in its healthy state. And you'll see both this pink stain, the Mason's trichrome, and then this myosin heavy chain red stain shows a lot of muscle tissue, as you might expect. Now, if you do an experiment where you cut out that muscle tissue and you just place it right back in after you cut it out, we see a modest amount of regeneration in that area, shown by the pink and the red stains kind of coming back. But if you do that same experiment, but instead of putting it right back in the same spot, you just rotate the tissue 45 degrees, we see much less regeneration occurring. And so what that's kind of hinting at us is the alignment within this tissue is super necessary for the cells to be able to regenerate after injury. Even cooler is we're now able to even see inside of the tissue. And so what we're looking at here is a movie of satellite cells, which are the regenerative cells in our muscle tissue. And you'll notice in this video, hopefully, that they're migrating from left to right primarily. They don't move that much from up to down. And so from this video, we have some evidence that the cells are relying on that matrix alignment when they are navigating the tissue space. So I want to again shift us over into hydrogels, which I'm going to introduce, and some work using 3D printing of hydrogels. So first, what is a hydrogel? Well, it's a water-swollen three-dimensional polymer network that starts to resemble soft tissues. And the reason why we might use hydrogels is because it's really good at supporting cells. It resembles the native environment that cells live in. And so we might use these for a variety of tissue engineering applications for soft tissues. And so if we broadly categorize hydrogels, there's two major classes that fall somewhere on the spectrum of bioactivity to tunability. The first being natural polymers. So those are derived from biological sources directly. They're kind of reprocessed, decellularized. And what I'm showing here on the left is an incredible work where we're able to 3D print, or not we, a group at Carnegie Mellon is able to 3D print a full-size model of the human heart using natural polymers, right? So we're able to create this macroscopic structure, which is amazing. The second class of materials are synthetic materials. And here's work from the Miller lab that used to be at Rice with some amazing abilities to model gas transport in our organs by 3D printing these hollow vascular channels. And so hopefully I've highlighted two major broad classes of hydrogels and some amazing works that's been done with those hydrogels. So the question then might be, well, you just showed me a bunch of cool stuff. What's there left to do? Now, I want to draw your attention to this diagram that maybe is a little bit confusing, but I hope to kind of break it down, where on the top axis, we're looking at feature scale, going at the most basic level from molecular all the way up to the macroscopic scale. And on the y-axis, as we go down, we're able to create more and more complex structures. And so if we look in the bottom right, we have the 3D bioprinting with a little E circle. And so, you know, with 3D printing, we're able to capture a lot of what we want to capture, but it doesn't extend all the way down to that molecular feature scale. And so what my goal is to kind of bridge that gap. And hopefully my hope is that by using materials that mimic the molecular and nanostructure of our body, we're then able to use these cell instructive cues that I talked about. And so again, in short, I want to recreate both tissue scale and the complexity. And the way that we're going to do that is this class of materials called self-assembling peptides that I could talk about for the whole talk in and of itself. But it's a class of synthetic materials that are built out of amino acids to form nanostructures that start to resemble the extracellular matrix in a lot of ways. And our lab rationally designs these structures as a soft hydrogel. And in total, for the rest of my talk, my goal is to fabricate macroscopically complex, hierarchically anisotropic, cell-instructive hydrogels to engineer tissues. And I'm going to break that down into two sub-aims. The first is trying to do all of that and use chemical cell-instructive cues to control cell growth. And this work was published over a year ago now in Advanced Materials, and this was my kind of first big paper, and it kind of blew up in a way that I didn't see, but I hope to kind of break it down for all of you here. And the second thing is using physical cues. And this was actually, for me, and I think Jeff, much more exciting if we can start to recreate the physical cues that cells can learn from and spread on. We can maybe work together with cells to build tissues. And this is going to be broken into two sub-aims, the first of which is currently under review, finally, after a very long time, and the second of which we're in preparation for, and I'm going to kind of present on all these three different things. So first, I want to talk about a specific type of self-assembling peptide, so that's a broad class, our specific class, which is called multi-domain peptides, or MDPs. And so they're designed in this unique structural way where they have charged amino acids at either side, hydrophobic amino acids on one face, and hydrophilic amino acids on the other face. And before I go into why have we designed it like that, one thing for me as an engineer that popped out very early on is this is an extremely modular system, being that we can swap out what those charged amino acids are on either end extremely easily. Our lab's done that for a long time. And so in this first work, I'm going to talk about two different variations of MDPs. The first, which I'm going to call K2, which has lysines, which are positively charged at either terminus, and the second called E2, which has glutamic acids at either terminus and is negatively charged. So we have a general structure that's similar, and we're going to basically just change the charge and see what we can do with that. So why are they designed in this way? Well, they were designed way before I got here, over 10 years ago now. When you place these materials into an aqueous solution, you see something very interesting that we like to call self-assembly occur, where one of the peptides will flip onto the other and create this hydrophobic sandwich. And this is all driven by non-covalent forces, which you may be familiar with, of thinking of them as like the weak bonds in our body. Well, it turns out that they're much stronger than maybe a lot of us realize. But when that occurs, you're going to have this kind of counteracting thing occurring, which we call molecular frustration that happens at the termini. Whereas if you have the same charge in close proximity, the molecule is not going to be very happy, and it's not going to want to kind of maintain the structure. But if you then add ions, similar to what's in our body in physiological solution, it is able to those ions are able to quench this molecular frustration, and we get this nice elongation of these fibers. And then macroscopically, when we look at this material on the left bottom inset, we can see this little hydrogel puck start to form that's on a syringe. And on the right side, I'm showing you scanning electron microscopy, which is extremely zoomed in version of our system. And we're able to see nanofibers starting to form. And so just to orient you to the size of the picture on the right, a cell would be much larger than this entire image. So when I was trying to basically break into Jeff's office and break into his lab, I was telling him, I think that this self-assembling nature of MDPs makes them really good for extrusion 3D printing. And I'm going to talk about why, but this has to do primarily, again, with this self-assembling nature of these materials. So this is going to lead me to my first aim, which I'm going to quickly kind of lay out for you, which is titled 3D printing of self-assembling nanofibrous multi-domain peptide hydrogels. So first, we did some materials and printing optimization. And then we were able to then create a series of complex three-dimensional structures, and finally use those 3D hydrogel MDPs for cell culture applications. One thing that I want to call to your attention really quickly is that we're doing this kind of in parallel with, again, that positively charged K2 and that negatively charged E2 material. And so before we jump into the fancy 3D printing stuff, we need to do some basic chemical characterization of these materials. And what I'm showing here is circular dichroism, where we can get an understanding of the secondary structure that these materials form, and the maxima at 198 and minima at 218 for both the K2 and E2 MDP show that we are getting a beta sheet secondary structure, as we kind of have seen in the past and would expect. And we can also do FTIR here to confirm that analysis. So again, we're getting these beta sheets starting to form. And then switching over to visually looking at the nanofibers, again with scanning electron microscopy, we can zoom in extremely high magnification. And on the top, we can see K2, and the bottom, we can see E2. And both of these form this nanofiber structure that looks kind of like a web. So now the question is, how do we print this material? Well, it has to have a very unique set of properties, because for these hydrogel materials, the way that we print them is we basically prepare them as a gel, and then we push on them really hard so that they flow as a liquid. And we design them in a way so that as soon as they stop flowing, they re-gel. And this self-assembly, again, of MDPs is what is going to allow this to occur. And so us as 3D printing nerds like to design some very basic structures to kind of test what can we build that is going to allow us to easily see if this material and printing system is going to work. So right here's what we call a 1x1 log pile structure with 1 millimeter by 1 millimeter pores. And I'm giving you a top view and a side view just to show that we build these up 10 layers tall to really test if we can 3D print, not 2D print like a lot of people do. And so if we just try and take our MDPs, we dye it blue so that we can see it, and we attach it to a 3D printer and start trying to print this structure, we're just going to get a hydrogel blob that's not very exciting or useful for many applications. And so now looking at a top view, right, that looks nothing like the design I laid out previously. And I want to point out two problems that we saw in this initial kind of work. The first is that the hydrogel was unable to support its own weight. And so that's a problem with the material itself. As it tried to go upward, it just sagged and kind of sank. The second problem that we ran into is that the printer actually wasn't optimized to print at the correct print speed. And I'll kind of dig into each of these two problems, both the material and the printing optimization individually. And so switching into a technique that we use for the material optimization, this is a beautiful schematic of a rheometer. And what this is designed to do is help us understand the properties of these gel-like materials. And so this is what we use to understand the viscoelastic properties of the materials that we work with. And just briefly, the way that it works is you have a gel that's then compressed between two plates, and then those plates will rotate back and forth. And by varying aspects of that rotation, we can understand the two main things about our material. The first being understanding the storage modulus, which we can think of as the elastic component of our material, and it's actually the energy that's stored in the bonds of our system as it's deformed. And the second being the loss modulus, which is the viscous component of that material. And you can think of it as the energy dissipated, usually in the form of heat, as you apply a kind of a force to it. And so the storage, again, to just kind of simplify, I think of it personally kind of like the solid-like component, and the loss modulus kind of like the liquid-like component. And so to kind of orient us to the data that I'm about to show, if the storage modulus, the G prime as we call it, is around 10 times the G double prime, then we have a gel. But if the G double prime is larger than the G prime, we have a liquid. And so the first thing that I did that blew my mind, that not many people had tried this before in our lab, but I took one of our materials, E2, and I just said, what happens if we keep adding more and more material to our system? So just very briefly, the way that we make these hydrogels from 1 to 4% is we take 1% our material, 99% HBSS buffer, or you can think of it as similar to water, but with a little bit of physiological salt added. And so if we just add the concentration of our material, what we see on this graph here, which is called a frequency sweep, is as we add material, the G prime on the y-axis is going to increase pretty modestly. And I just want to call to your attention that the axes in these graphs are on a log scale. So a small difference is probably much bigger than you might think. And so if we run this same sort of experiment with K2, we see that we're able to kind of achieve gels that are much stronger than what we had previously seen in our lab by going up to this 4 weight percent concentration. And so I got very interested in this 4 weight percent concentration because when I was working with this material, I was like, this is able to kind of hold its structure itself. Can we validate that this material is going to be useful for 3D printing and meet some other things that are necessary for what it takes to be 3D printable? So I'm going to break that down into 3 separate slides with 3 properties. The first being a flow initiation. And this is a property where if you apply enough strain to your material, if you push on your material far enough, basically, the material should start flowing at a certain point. And this is important, and this property and the next one are also very important for a lot of food products or toothpaste, or think of ketchup and mustard in a bottle. If you squeeze on that bottle, you want the ketchup or mustard to start flowing out. And so what we're showing here on the right side is we're increasing the strain, so the amount that we're deforming our material. And at the beginning of the graph, we're seeing the G prime and the G double prime. And the G prime is over 10 times the G double prime, showing that we have a nice gel material. But as we increase the strain from left to right, we get this behavior where the G double prime actually will jump ahead of the G prime, and we get the transition from a gel to a liquid. The second property that's also very similar to this first one is that our material is shear thinning. This is a property that's also important for a lot of food products. And in this test, what we're doing is we're basically spinning the parallel plate faster and faster and faster, and we're measuring how viscous that material is. And what we want to see in this test, that it has this negative slope. And just to kind of root this in our it's hard to think about these material properties a lot of times, but what I think of this is, it's the opposite of quicksand. So quicksand, if you try to pull, it's going to get much harder. this is the opposite. If you try and pull, it's going to get easier and easier. And the third property that I would argue is maybe the most important property is that our material is what we call rapidly self-healing. And so in this test here, what I'm showing is oscillatory strain sweep, which basically in these white regions is applying a very low strain. And then in the gray regions, I'm applying a bunch of strain. And so as we shift from the white to the gray region, we'll notice that the red lines will invert. And so what we're seeing is basically the liquefaction of our sample as we apply that super high strain. But the most important point is actually that interface between the gray and the white regions, where we're able to see within a matter of seconds, our material is able to recover all of those gel-like properties. Now, I'm showing some pretty graphs, but I think it's much easier to see this in a video. And so what we're actually seeing here is our material is able to liquefy and then instantly recover its hydrogel properties and self-support over these long overhangs. So this was probably the most exciting videos that I got maybe in my PhD, because once I got this, I knew kind of everything else was going to work. And yeah, I just want to highlight that again, that I was ecstatic. Let's just leave it at that. So after we've now optimized our materials, the next thing that we need to flip over to is optimizing the printing conditions. And so there's going to be kind of two bad scenarios and one good scenario. And the analogy I like to use for this is putting toothpaste on your toothbrush. So if your arm is moving too slow as you're applying toothpaste, you're going to get like a fat blob of toothpaste that's maybe going to get in your way more than it is useful. And if you apply that toothpaste, but your hand's moving too quickly, you're going to get breakage of the toothpaste and not enough toothpaste to be deposited. But if you're able to get the exact right speed, you can get these nice even distributions of your line width. And so what we did in this experiment is printed a series of lines at faster and faster speeds. And on the y-axis, we're plotting the width of that line that's being extruded. And on the x-axis, we go left to right, we're going faster and faster. We can see this asymptote start to form, and we're reaching this plateau where our line width can be kind of predicted based on our print speed. And so I just want to quickly call to your attention that the ideal line width that we should be able to achieve is the inner diameter of the nozzle that we're printing out of. And in this case, it's 250 microns. But unfortunately, we're not able to reach that kind of ideal case before we see line breakage. And that's because our hydrogels that we're printing are not actually going to be perfect cylinders, but they're going to be sagged a little bit. And so the height is going to be shorter than the width. And we're going to be kind of limited in that sense. And this will be a concept I come back to much later in the talk. So once we do that optimized material and we do that optimized printing, we can now build complex three-dimensional shapes shown in that video here. And I've done plenty of 3D printing, but I still find watching videos of 3D printing to be sort of mesmerizing. And also for all of us here, I have some examples of this structure just to kind of give you a sense of scale that I've saved for over a year now. But to kind of show some other designs that we went after, we started with the cylinder. And to orient you again, a cylinder was the most complex structure that had ever been printed with a self-assembling peptide. Well, we're able to print a cylinder, and then again, we're able to do a 2x2 and a 1 log pile that were each 10 layers tall. And even more so, we were able to then mix in, so I should have mentioned this earlier, but the orange dye is just a dye that we add to our material to kind of identify it, and that's our K2 material. But the blue dye here in this far right print is our E2 material. And so what we're able to show is we can start to create multi-material prints, which again is the first demonstration that this has been done with a self-assembling peptide system. So next up, what do these materials kind of look like? Well, I just have a quick video here to show you what they look like. They look a little edible. I'm sure Jonathan in the back there has eaten one of these before. And you can kind of pick them up out of solution, and this makes them useful for cell culture applications. So we then went ahead and switched our gears to, now that we can print, what can we use this for? And again, what I'm showing on the top row, just to orient you, the top row is our K2 positively charged material. The bottom row is our E2 negatively charged material. And you'll see these big kind of holes in these images. Those are actually the pores in our 3D printed structure that we saw previously. And then we're doing live dead staining onto C2C12 myoblast cells. And so we basically take a myoblast cell line, we put them onto our scaffolds, and we're looking at them at day 1 all the way up to day 10. And so these little green dots are individually each live cells, and red dots, which you won't see a lot of, are dead cells. And I want to point out 3 main things that we observed using our system. The first is that we got high cell viability regardless of whether we used K2 or E2. Independently, up to 10 days, the cells were surviving and happy for what we could tell. The second thing that we noticed is that while cells were growing on the negatively charged E2 material, the cells were growing at a much faster rate on the positively charged K2 material. And the third thing that you might notice in the far right image, comparing the 2 right images, is the cells on the positively charged material are kind of all like spread out, whereas those on the E2 negatively charged material are kind of bundled up. And to very quickly tell you why we think that's the case, we think that it's actually an electrostatic interaction between the negatively charged membranes of the cells. And so they're naturally kind of pulled on our positively charged material and feel pushed on our negatively charged material. And this is also kind of like seen in a lot of cell culture applications where we might use poly D-lysine coated plates. Well, K2 is a poly D-lysine mimic, if you want to say that. And so then we went ahead and took some images now where we're doing confocal microscopy of on the blue channel, we have the DAPI, so that's the cell nuclei, and on the green channel, we're looking at the actin filaments of those cells. And again, the cells are spreading out on our positively charged material, but we also saw something very exciting shown here, these 2 tube-like structures. And what we think was happening here and got me really excited about kind of moving this project to the next stage is we started to see some what we think is differentiation of those cells. So the myoblasts will naturally differentiate into myotubes. And what we were thinking is without us biasing anything, that seems to be starting to happen on our structures. So to summarize AIM-1, the self-assembling nature of MDPs makes them really great for extrusion 3D printing after a little bit of optimization. And we were able to do that optimization for 3D printing and then create a series of complex structures and multi-material prints. And then finally show how we could use MDP charge to use that as a cell instructive cue and control cell growth. So to kind of orient us back to what we want to complete, we were able to get macroscopic complexity and we're able to get cell instructiveness. But are these hierarchically anisotropic? The answer is no. If we take a zoom in on the nanofibers within this system, contrary to what I expected when I actually was starting to do the study, I thought that the fibers would maybe start to align, but we see no kind of nanoscale fiber alignment on our system. And so the next kind of pursuit was to create macroscopically complex, hierarchically anisotropic MDPs. When I say, I just want to quickly clarify, when I say anisotropic, because maybe I didn't talk about this earlier, anisotropic is a property where the properties in one direction are different from another direction. So if we think back to the skeletal muscle tissue, you can think that in the aligned direction, the properties are going to be different than the kind of radial direction. And that's going to jump us into AIM-2A, which is being able to accomplish fiber alignment with our MDP system. And so this is going to get a little bit confusing here, but I hope to break it down for all of us. In AIM-1, in all of that work, we were using a gel, a hydrogel. And from now on, I'm going to call that pre-gelled. And again, what that was is it was an MDP dissolved in a physiological buffer that creates a hydrogel. And there is some mechanistic understanding from another self-assembling peptide system here, where when you apply a shear force, you don't actually see fiber alignment. And there's a hypothesis that what happens is the macroscopic nanofibrous structure will break down into these smaller domains that are small enough to flow. And unfortunately, if you kind of shear them and stop shearing them, we will not achieve fiber alignment. So the key to this work was able to achieve fiber alignment by basically decoupling the gelation process and the application of shear. And so what I'm going to introduce in this AIM is this new thing called a precursor solution. And this is our MDPs, but not dissolved in a buffer, but just dissolved in water. So it's going to have liquid-like properties because it doesn't have those ions yet to kind of quench and convert to a gel. And I'm going to show that here kind of in green. And secondarily, we're going to introduce this other idea of a gelation bath that has counterions. So again, on one side, we have our precursor. On the second side, we have those counterions. And if we apply a shear force onto our precursor solution into that gelation bath, we get the transition from a liquid to a gel. So I'm going to show a quick movie of that happening. Again, our precursor solution is dyed orange just so we can see, and we're able to create, just using a standard pipette tip, some really nice looking MDP noodles. And what was actually most exciting about this work, because we didn't really know any of this stuff yet, is that we could actually pull these little hydrogel noodles out of solution. Now, why this was so surprising for us is because we're, again, working with a supramolecular self-assembling system. And so within all of this, there are no covalent bonds holding this noodle together. So I went ahead and did a series of screening studies using a technique called polarized light microscopy that I want to just quickly try to give a quick explanation of what we're looking at. When you take white light and you place it through a polarizer, similar to polarized sunglasses, that light is going to be polarized into one plane. And if you have a cross polarizer in front of that, that's basically polarized orthogonally, you're going to cancel out all of that light. So if you have sunglasses on that are polarized, and where I've seen this mostly is when I go into like a boba shop and I look up at the screen and I have these sunglasses on, I'll notice, wait, the screen got super dim. And especially if I start to rotate my head, the dimness of that screen will change. And that's because you're basically cross-polarizing with your own head. But if you're able to put a material that's anisotropic in between those cross polarizers, it's going to kind of split that wave into two different planes. And those two different planes are going to be able to cross through that second polarizer. And so when we look at these images, the brighter the light that we're able to observe, it's more suggestive that we have anisotropy within our material system. So now I have a video of a hydrogel being created with a pipette tip, but this is being videoed through a polarized light microscope. And so this is the noodle that's being formed, and you might see these bright areas of the noodle, and that is suggestive that we're seeing some sort of alignment in our system. So using this technique, I went ahead and screened a series of first precursor solutions, starting at 1 weight percent K2 dissolved in water, all the way up to 4 weight percent K2 in water. And what we saw is that 3 weight percent K2 seemed to be kind of the perfect point where we get nice polarized light coming through, and also a noodle that looks like a noodle and not like a blob in the 2 weight percent case. And so because of that, the remainder of the study was done at 3 weight percent. The next thing, and I'm going to throw a bunch of images at you really quickly, is doing a screen of a bunch of different conditions for that second aspect, the gelation bath. And I want to just quickly orient us to this region, which is what I care about and got interested in, was we saw pretty different properties between the left and right blocks in terms of the handability of these materials. And this region is interesting to us because this is what 1x PBS, that's commonly used in cell culture, so phosphate buffer saline, and 5x, that phosphate buffer concentration. And so now that we'd kind of qualitatively screened a bunch of gelation baths and a bunch of precursor solutions, we did a much bigger deep dive into this. So in all the left side, I'm going to have our pre-gelled solution, which again is from AIM-1. That's our negative control, where we're looking at the top, an image of polarized light microscopy. So there's maybe a little bit of brightness. And then at the bottom, we're looking at scanning electron microscopy. When you zoom in on that structure, there doesn't seem to be any sort of alignment. But if we introduce 1x, oh, this is in the way. Let me just move this. Okay, so if we introduce 1x PB, and that's just corresponding to PBS buffer, as you may buy or use commonly, we start to see birefringence and also some slight alignment if you squint and look at it at the right angle in the scanning electron microscopy images. And then because we were interested in kind of this region, we are going to introduce these two new gelation baths, which are 3x phosphate buffer So this is PBS, but you just triple the amount of phosphate buffer. You don't change the salt that's going on in there. And then the 5x case where you're 5 times the phosphate buffer concentration. And what we're really able to see is through polarized light microscopy, and then also by directly observing the fibers, an increase in that alignment and that birefringence. And so we were able to show that with K2, we're able to get this tunable, this ability to tune the nanostructure. And that was super exciting for us because this is the first time that anyone's been able to tune the nanostructural alignment within a self-assembling peptide system. So next, after all of those qualitative assessments, we turn to a quantitative technique called 2D small angle X-ray scattering, where you're basically observing the diffraction pattern of shining X-rays at your specimen. And so these bright images on the top are just what that pattern looks like. And at the bottom, I've graphed as a function of the angle around that diffraction pattern, what the intensity of the signal is. And so again, as we go from left to right, we'll see an increase kind of in that bottom quantification of this pattern. And in the top, it'll look less and less like a circle and more pointed. And so again, this is quantitatively showing us that this alignment that we thought we saw is actually the case. Next, I'm going to switch over to the properties of these hydrogel noodles, starting with the 1xPB noodles. And what you may notice is that I'm trying to pull these up out of solution, but they're kind of trying to fight back against me and pull themselves back into solution. And what we're actually seeing is that the strength of these noodles is not enough to self-support over these long distances, and they're actually fracturing. Now, this is in stark contrast to the 5xPB noodles, where I'm able to easily pull them up out of solution. I'm able to drop them pretty aggressively, and they're able to maintain their shape. And I can kind of repeat this over and over. And to quantitatively assess this behavior, we turn to this technique called optical coherence elastography, which here's a full schematic of the system, but I want to draw your attention to the far right part. But before I get into that, why are we using this technique as opposed to just rheology like I showed in the first aim? Well, rheology is not designed for anisotropic materials. It's designed for materials that act the same in every direction. And so because we have an anisotropic noodle system, what you would usually turn to is some sort of tensile testing or some sort of poking testing. But for our weakest of noodles, they aren't really strong enough to carry out any of those testing. And so what this technique does, it's a non-contact method of assessing the mechanical properties. And so what's done is a wave is basically shot through your sample, and you're able to observe that wave as it propagates through your sample. And you can correlate the wave at which the speed is moving with the stiffness of your material. And so I'm going to show a bunch of videos on the right here from pre-gelled down to the 1x, 3x, and 5x case. And what I want you to realize, in addition to kind of the colorful nature of this method, is how the waves are moving faster and faster the further you look down our materials. And so we can quantify this data as the wave speed as a function of our different material systems and see some pretty drastic differences in the wave speed. And just to orient us, like what does this look like in terms of the tissues in our body? Well, first, I just want to say that we're going to make some pretty big assumptions that we know are not actually the case to be able to do this, where we're going to assume that our system is isotropic, so that this anisotropy doesn't actually happen, to be able to get the bulk stiffness understanding of our material. And so in this kind of spectrum here of different stiffness values, our pre-gelled and 1xPB noodles fall somewhere close to brain tissue, whereas our 3xPB noodles fall close to muscle tissue, and our 5xPB noodles are approaching bone tissue. Now, I do want to again emphasize that this is not an apples to apples comparison because we're again assuming that the strongest part of our system is the strength in every which direction. But it is pretty cool that without changing our material, we're just changing the way in which we're extruding and processing our material. We're able to get this range of mechanical properties. So then switching over into the application of this system, we went ahead and used this cell line called valvular interstitial cells, which are derived from heart leaflets within pigs. And we did a study where we looked at them at day 1, day 3, and day 7 on all of our different hydrogel cases. So what I'm showing on the left is a confocal microscopy image at day 1, day 3, and day 7 of these cells. And And on the right, what I'm going to show, what I'm going to show on the right here is a Fourier transform that's been performed on the image to basically unbias our eyes and show us, is there alignment in the system? And the more kind of aligned that little graph looks, it's a assessment of the actual alignment in the image. And so when we do the same test with our 1xPB hydrogels and also our 3xPB hydrogels, we start to see very nice alignment of the cells along the direction that we would expect. And then here's kind of a big curveball that I'm going to throw to you that took this paper in a completely different direction, which was in the 5xPB case, where we might expect the highest alignment of our cells, we started to see that actually the cells by day 7 were aligning no better than our negative control. And so just kind of to bring us this full picture, we're looking at the top at the nanofibers of our material, and at the bottom, the cell behavior. And again, that we weren't seeing this spreading as we might expect. So before I talk about some of maybe why we see that weird behavior on the 5x case, I want to just show off how cool this really is. We're using our 1xPB noodles. So this is a macroscopic picture of these valvular interstitial cells at day 3, and at day 7, they're just kind of rapidly growing to confluency, and they are all aligned, or mostly aligned, in the direction we would expect. The next thing that we did is we switched over back to those myoblast cells that I discussed in the first aim, and we saw, could this be useful also for muscle tissue? And what we're going to look at here, this green stain is not actin anymore, but we're actually looking at a differentiation marker of those of myotubes. So this is what I kind of alluded to earlier. Now, anything that's stained that bright green color is a myotube. And so on the pre-gel case, you see a bunch of myotubes going in every which direction, which is not that useful for tissue engineering applications, I would say. But on our 1 and our 3x case, we start to see the emergence of these aligned myotubes in the direction we would expect. And again, we're not adding any sort of external factors to help this process occur. So we think that this is like an unoptimized case. Now, to switch back over to why do we see this behavior on the 5x hydrogels, we switched back to scanning electron microscopy. And what I'm showing you here is an image of, in gray is actually the hydrogel nanofibers, and then falsely colored in red and in blue. And falsely colored means I basically colored it myself in Photoshop to help us in visualizing what's going on. We can start to see the cells interacting with the nanofibrous structure. So in the 1 and the 3x case, what I like to think of this behavior when I look at it is we can see the cells kind of pulling those nanofibers apart. And then I think that's most clearly seen kind of looking at the regions close and far from the cell, where the fiber alignment looks pretty drastically different. Now, in the 5x case, we see a totally different sort of cell morphology. We don't observe that kind of pulling behavior of the nanofiber system. And so, again, this is a hypothesis. We think that the fibers are so aligned and so packed that the cells can't really tell that this is not just a two-dimensional surface. So to orient us back to what we're trying to achieve, did we get hierarchical anisotropy? The answer is yes with these noodles. And did we get cell instructive cues? The answer is yes. But did we get something that's macroscopically complex? The answer is no. So we went forward in one aspect and we went backward in another aspect. And so the final part that I want to talk about is, can we create macroscopically complex, hierarchically anisotropic MDP hydrogels? And so what we turn to is this technique called support bath extrusion printing, where when you work with liquid-like materials and you want to print them, you can either do the materials optimization that I talked about in aim 1, or you can use this fancy technique where you're actually printing your material into a gel in and of itself. And so what that enables you to do is support as you print your structure until you're done printing. And then in this case, the temperature is turned up and the melting of that gelation bath is able to occur, or the support bath is able to occur, and you can get this kind of macroscopic structure. And so what I wanted to do was basically take this technique and append it to what we just showed in aim 2A and say, can we use this to create aligned macroscopic structures with our MDPs? And so before I dive deeper into kind of how we did this, the first thing that we need to consider is what are the possibilities that can happen with the support bath? Well, if we have a support bath that's too liquid-like, kind of similar to the solution we were extruding those noodles into, you're going to get your material not forming nice fibers, and you're going to get some breakage. Whereas if your bath is too solid-like, your material is actually going to flow in the wake that's created when that bath is deformed. But if you get the perfect kind of bath that matches your material properties, you're going to be able to create these nice fibers when printing. And so there were 3 support bath candidates that we went about tackling this. And I just want to shout out that Jonathan did a lot. Jonathan in the back there, my undergrad, did a lot of the brunt work in terms of building these baths and testing them all out. But there were 3 candidates, the first being what we call fresh version 1, the second fresh version 2, and the third being Agros. So fresh version 1 was a paper out of Carnegie Mellon that is the same group that printed that full-sized human heart, where they took a gelatin block and they just used a commercial blender and blended up that gelatin into these kind of microparticles, and then use that as your support bath as you're printing. The second is an upgrade on that, where instead of using a commercial blender, they're doing a little bit more of a fancy processing system to create these uniform particles that you can then use as your support bath. And the third is Agros, which is commonly used for 3D printing of itself, actually, or in a lot of other molecular biology labs. So what I want to just quickly orient us to is we're going to, again, turn back to that rheometer from the beginning and look specifically at the viscosity of our material shown in red versus the fresh 1, fresh 2, and Agros baths. And what we would expect and what we actually see when we go ahead and print is that the material, that the support bath that has the closest viscosity to our material, leads to these nice smooth fibers. Whereas the fresh version 1, which we were hopeful would work, had these kind of huge ridges, and when we would melt away that bath, it would actually just disintegrate our print in total. So then we went ahead and did some print speed optimization like we did in the first game again. We're basically printing a continuous line by moving faster and faster, and we can again measure the line width. And what I want to call your attention to back is this ideal nozzle diameter of 250 microns. In our old system, we couldn't get anywhere close to that. But in this new system, we're actually able to get to that ideal nozzle inner diameter of our line width and even underneath that. And I know that's a little bit weird, but basically we're able to get resolutions that we were unable to achieve in the past. And so combining this with what we showed in 2A, we tried to print into the same Agros support baths, but in one case with 1x PB, and the second case in 5x PB. And when we did SEM, we saw exactly what we were hoping, where we saw these differences in the nanostructural alignment based on the support bath concentration that we used. And then kind of pulling this all together, and probably in the most exciting time of kind of starting where we are at the very beginning and getting to where we are now, we're able to print 3-dimensionally macroscopically complex hydrogels that when we do SEM on them are hierarchically anisotropic, and also when we plate cells on them are also cell instructive. So to summarize aim 2, we fabricated K2 hydrogels with a spectrum of fibrous alignments, and we found that higher alignments led to stiffer matrices. And we showed that this alignment was able to direct cells in the 1 and 3x case along the direction of alignment that we hoped, but that cells we hypothesized that aren't able to mechanically couple were not able to align. And then finally, we're able to use this support bath 3D printing technique to build 3-dimensional structures using what we learned. So with that, I'd like to thank all of the HeartDrink Lab. You guys are an amazing lab to work with, and I'm really thankful that I got to spend my PhD with all of you guys. I know that sometimes I can be a little bit annoying or grumpy, but thank you all for putting up with me. Second, I want to just thank Jeff so much for one, trusting me when I told him that I could figure this out. I really, really appreciate that. And B, putting up again with me as I go through my highs and my lows. And also, I would like to thank my thesis committee, Dr. Grande Allen and Dr. McHugh. Thank you, Dr. Grande Allen, specifically for keeping me here at Rice when I almost left. And I just want to give a couple of other quick shout outs. Thank you for Booty for all the microscopy help. Thank you for Uday for help in the biomaterials lab. And thank you also to Jordan Miller for also being a large reason why I found the HeartDrink Lab at Rice. So I'm going to now kind of shift to the cute picture part of my presentation, where I'm going to show you kind of my motivation for doing all this work. I just want to thank, as Jeff alluded to, teaching was very difficult. And I just want to say that all of my work that I do is driven from my experience. And I have a funny picture to kind of lighten the mood a little bit of Jack after he cut my hair, and a fun video of me kind of dancing with my students. But I'd also like to thank all of the wonderful friends that I've met here in Houston, either in or outside of lab. I'd like to thank my friends that I knew before Houston that have come and visited me in Houston, and all my groomsmen, and then also the Manso clan that I've happily joined. And then finally, my family that I've kind of grown up with all my life, featuring the newest addition in the upper right corner. And then the final three kind of shout outs I have is one for my parents. Thank you guys for always being there for me while I was venting over the phone, or explaining to my dad the fibers aligning versus the cells aligning versus the fibers aligning over and over. And then second to last, we have my wonderful wife Shannon, who's just been one, financially supporting me my whole PhD, which has been great. B, emotionally supporting me my whole PhD, and just brought me so much joy and happiness in my life. And I love you, Shan. And getting married was a great day. I can't think of the last great day like this. And then finally, I have Jeff's favorite part, the pictures of my dog. So quick side story. I remember very early in my PhD, Jeff was like talking to me about how he thinks it's so crazy how people would treat their dogs like they're children. And I was like, yeah, that's totally crazy. As in the background, I'm treating my dog exactly how I'd treat my child. So this is my dog, Marshall. We got him right after I got to join a lab. We decided to get a lab together, and he's been through a lot of highs and a lot of lows, and it's really showed me the priorities in life. And then finally, I have a montage of videos, his favorite thing to do, which is eat either pup cups or his puppy patties when we're having a fun little time. So with that, I'd like to open it up to if anyone has any with me for.
Question from